So here we can think about how does a neuron decide to respond to reception of EPSPs and IPSPs. In this illustration, your EPSPs are shown in green, IPSPs are shown in red. Now, what we really start to focus on in order to figure out if we are able to elicit an action potential is summation. So just meaning sum, adding everything up. So thinking about the total amount of signals that a neuron receives, and that's going to impact whether the combined event will be something that's exciting or something that's inhibiting. So a single EPSP is really not enough to elicit an action potential in a postsynaptic neuron. But if there are many excitatory terminals that are firing on that membrane of the postsynaptic cell, or if the rate of the delivery is fast enough, then the likelihood of reaching threshold increases, which would mean the likelihood of an action potential occurring increases. So if you have kind of an overwhelming stimulatory effect, then the membrane may have enough stimuli to reach that threshold. So what we see in A is an inability to achieve that. So if we don't reach threshold, we have no summation that's taking place here. So notice that we had a little EPSP here, and they weren't enough, right? And there was no summation. It might have just been that they happened too far apart from one another in order to add them together. Now, if we are incapable of achieving summation or reaching threshold, then another way of describing this process is that the neuron is facilitated. It won't fire an action potential. But as we can see, it would be a little bit more easy if we have an event that happens really close or really consistently behind this. It might be easier to depolarize because it's closer to threshold. But if we wait too long, then we're back to resting state. So it won't be any easier. Now in B, we have temporal summation. So temporal, you're thinking of the word time. So in temporal summation, we're adding things up, and this is based on rapidly firing presynaptic neurons. So we're yielding a ton of EPSPs that are close together with respect to time. And the summation of these can bring the initial portions of the axon, so we're at the axon hillock here, to the point of threshold. And so temporal summation, the way to think about it is if you're telling someone to do something and just one person is talking to you and they're just going, go, 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 go. They just keep saying it over and over again. And then enough of that stimulation, when you're adding it all up, is going to say, all right, I'm going. Okay, so that's your temporal summation. Now, spatial summation, we're thinking about space. So in this scenario, we have more than one presynaptic neuron firing at the same time, and we're at different locations on the neuron. So at the same time, we're generating an EPSP. So we add that up and we can also reach threshold. So this would not be the same idea as what I've said before. So notice that while it says E1 and E2 up here, notice that the illustration is just showing you E1. E1 is talking, go, 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 okay? Now imagine that you have two different EPSPs in two different locations and they're both yelling at you to go. Okay, so they're not saying it a ton of times, but now you have more individuals yelling that same message at you. So that's the distinction between the temporal and the spatial. Now, when we think about this idea, again, you can have EPSPs and you can have IPSPs. And there are times in which an EPSP and an IPSP can cancel each other out, in which case even the presence of the EPSP is not sufficient because we don't quite reach threshold. Okay, so we can have spatial summation of factors that are opposing one another with respect to the magnitude of that membrane potential. So when it comes to IPSPs, if we're trying to prevent a particular response, they're most effective when they're located between excitatory inputs and where an action potential will be generated, because they're kind of like, I'm initiating something, I'm telling you to go, and then all of a sudden an IPSP is like, no, don't go. Okay, so they're really preventing that signal summation from yielding threshold and therefore resulting in that all or nothing event. As it's important to initiate this process, it's equally important that it ends. So we want to consider the termination of neurotransmitter signaling, because that presence of a neurotransmitter can elicit a downstream response. So after a response is triggered, the chemical synapse needs to return to a resting state. So we want to ensure that there are no neurotransmitter molecules found at the synaptic cleft anymore. So with that said, one can imagine, that anything that blocks the process of termination from occurring can have significant impacts. Now, something that is discussed within your Campbell book, and I think this is in the enzymes chapter, is the role of biological warfare and sarin gas and how it can trigger paralysis and death. Now, the focus at that time was more on enzymatic inhibition and the irreversible nature of this particular example. So there was DIPF, which was an irreversible inhibitor of acetylcholine esterase. So at this point now in the semester, you've had a lot of exposure to acetylcholine from cell communication, and we can revisit this concept and think about its role in terms of allowing normal function of the nervous system to take place. So if we have an irreversible inhibitor of an acetylcholine esterase, okay, and so that helps to remove acetylcholine concentrations at the synaptic cleft, then what ends up happening in that scenario is a ton of downstream responses that are inappropriate. So we just have too much. And so that can lead to, uncontrolled skeletal muscle movement. And so this can eventually cause death. And this is why it was being utilized as a form of biological warfare. And another way to think about this in terms of neurotransmitters and their presence and their removal can be that of psychoactive drugs and the role that certain types of substances can play within our body as it relates to the nervous system too. So we know from our discussion of cell communication that if we turn on a response, then eventually we want to be able to turn it off, right? We don't want to always elicit some type of downstream cascade because this can really defeat the purpose of achieving homeostasis, right? And balance is something that we ascribe to the nervous system and the endocrine system. So for signal cessation, which is essentially what this is, we have to think about how do we turn off this response so that we don't have prolonged communication. And the stimuli in this scenario, because we're focusing on the chemical side, is the neurotransmitter. So one easy solution would be, if we don't want to elicit a downstream response in a postsynaptic neuron, stop releasing the neurotransmitter, right? And so we can say, let's just stop firing that presynaptic neuron. We have other options that are available as well. So we can have the release of neurotransmitters and their subsequent diffusion away from the synaptic cleft. That's not visualized here. And they're just kind of floating away into the extracellular fluid. Now, the neurotransmitters that do this may be reabsorbed by supporting cells, so glial cells, and returned to the neurons. Another option, which is shown in portion A, is enzymatic breakdown of the neurotransmitter, so degradation. And that degradation event takes place within the synaptic cleft. So we have the breakdown of products that were released by the presynaptic neurons, so our chemical messengers. And when we do that, they're no longer capable of eliciting a response at the postsynaptic neuron because we know that ligand binding is specific. If it doesn't fit into the site, we don't elicit a response. So when it comes to these neurotransmitters that are degraded, here's kind of showing it, and this kind of process takes place through an enzyme, right? But once it breaks it down to smaller components, it kind of just shows it heading off to the side. It doesn't always have to be the case. It is possible for degraded neurotransmitters to be reabsorbed by the presynaptic neuron, and they can be recycled. So they can be synthesized in order to generate more neurotransmitters that can be utilized when they're needed. Also in this image, it's showing you the inactivating enzyme present on the postsynaptic neuron, but that enzyme may also just be present at the cleft. Okay, so that's panel A. Now, you can also have reuptake. So reuptake in the absence of enzymatic degradation. And so the synaptic knobs can reabsorb the neurotransmitters in their state that they are, and they can simply be stored as shown in the illustration, or you can degrade them once they are back in the presynaptic neuron. So in looking at these examples, just think about how this can help to stop any downstream communication that you're trying to avoid having. So this gives us a sense of how we can elicit a signal, how we can propagate that signal, how we can transition from something that's electrical to chemical, how that chemical release can influence what happens in the next cell, and what we would need to do if we no longer want that process to take place. So I really want you to think about how this relates to how your semester essentially started, right? Because cell communication was really early on. Think about a lot of the things that are similar in our discussion or in our expectations of cells that are found within a multicellular organism, and really start to apply them to the nervous system. So I often put the nervous system at the end because it gives us a good sense of all of the concepts that you've already learned and a way to piece them together, which makes it a lot easier at this point in the semester for you to think about the content more critically.
Neurotransmitters, along with electrical signals, are essentially the language of the nervous system. So this is the means by which neurons are able to effectively communicate with the rest of the body. So things like sleep, hunger, rage during a lecture exam, these are all due to the actions of these molecules. And there are many neurotransmitters that have been identified, and they can be grouped based on their molecular structures. And here is only just, a select showing of neurotransmitters. But when it comes to the list that we have here, there is acetylcholine in a list all by itself. The first identified neurotransmitter, it was found at neuromuscular junctions, which are really much easier to understand as compared to events that might take place deep within the central nervous system. So there was a lot that was elucidated from acetylcholine. We've discussed acetylcholine esterases and their role in clearing the neurotransmitter. So this is just one type, right? And it's also one that was mentioned within the cell communication chapter. There are also amino acids. So the name in and of itself says what you have here. There are biogenic amines. These are synthesized from amino acids, but there's the removal of the carboxyl group. So as a reminder here, it would be a carboxyl group, but there is the retention of the amino group. So they'll have an amino group, but no carboxyl group. And there are neuropeptides. So neuropeptides can be 2 amino acids long or up to 40 amino acids long. And then there are gases, an example, nitric oxide, which was discussed within the cardiovascular system portion. And this is like an inorganic exception to the usual definition of a neurotransmitter when we look at all the examples that are seen above it. And when it comes to gases, these are synthesized as needed rather than stored within synaptic vesicles. So they'll diffuse out of an axon terminal rather than be released through exocytosis, and they'll diffuse into a postsynaptic neuron rather than bind to a surface receptor. And while there's great diversity in the neurotransmitters, it's important to keep in mind that the function of a neurotransmitter is determined by the receptor to which it binds. And so that statement brings us full circle to the start of the semester in which cell communication was covered. When it comes to this particular image, there isn't a need to memorize these neurotransmitters, meaning you're not going to see a structure and be asked to indicate which neurotransmitter is it. The purpose behind having this slide is just that you see some examples of neurotransmitters. Think about how we categorize them. Know that the list is growing, so our understanding of neurotransmitters is constantly evolving, and just that the presence of a neurotransmitter is not enough to say what will take place. What also matters is the receptor that it interacts with. So examples of which would be like GPCRs, enzyme-linked receptors, things of that nature.